Hello, everybody. My name is Oleg Tsodikov, and I'm a professor at the University of Kentucky College of Pharmacy. And today I'll tell you about agents that are called Mithramycin derivatives that my team and I are developing for treatment of children's cancer. And this cancer is called Ewing sarcoma. And when diagnosed early and when it's localized, it's fairly treatable with surgery and radiation, but when it's spread, when it's metastasized, or when it recurs, the chances of survival for these patients are very low. And the treatment is, in addition to surgery, consists of toxic, old, and non-precision chemotherapeutics. And you can see here is a bone eaten away by a cancer tumor, by Ewing sarcoma cancer. These cancers are fairly unique in that they're characterized by mostly one mutation, and that's a abnormal genetic fusion called EWS1 or EWS-ERG. And that's an abnormal transcription factor that has been deemed undruggable. It's a DNA binding transcription factor, and it's similar to transcription factor that drives aggressive prostate cancers that also spread into the bone, where the transcription factor here is sharing this ERG DNA binding domain with Ewing sarcoma. The agents that we're developing are semi-synthetic analogs of natural product called Mithramycin that we make by fermentation in bacteria, much like one brews beer, and then extract these and chemically modify them to attach this moiety, these different moieties that act as an antenna and interact directly with the transcription factor that I mentioned before, the EWS1. We know in atomic details how these side chains interact with the target. In cell-based assays, unlike parent drug Mithramycin, these analogs are highly selective, and basically the selectivity is, you can look at it as separation between the red line, the activity against the Ewing sarcoma cells, and the blue line, the activity against other cells. The higher the separation, the higher the selectivity. So you can see some of our analogs are highly selective. They're more than 20-fold selective. And in addition to cell-based experiments, this selectivity translates into the mice models of Ewing sarcoma, the xenograft model, where the tumor is grown in the mouse, and in the absence of drug, the tumor grows 20-fold in 10 days. And in the presence of one of our analogs, tryptophan analogs, the tumor growth is delayed for up to 60 days. And in terms of survival, all mice that are not treated die within 13 days, whereas treated with this tryptophan analog, all mice stay alive for up to 80 days, and some mice stay alive for up to 100 days. These analogs have been patented by us. So we have both full patents, issued patents, and the pending patents. Currently, this project is sponsored by NCI, R1, multi-PI-R1 grant, and we're extremely grateful to the NCI for this. But we are looking for partners to carry this project to the bedside, and we hope that some of you will want to join our team or will want to license the technology to help save these unfortunate kids that have this cancer. And so right now we're in the middle of the R1 project, where we're developing proof of concept and doing some IND enabling studies. And basically at the point where we're ready for some more advanced IND enabling studies, and thinking about narrowing down to one clinical candidate of 2 or 3 lead analogs that we have right now. And moving on, of course, we're hoping this can progress further to phase 3. And our team is myself. I do basic science, structural studies that you've seen in one of the slides, and drug design based on these structural studies, as well as biochemical studies. Jürgen Rohr lab are making these molecules, growing bacteria and Mithramycin, modifying Mithramycin. And Mark Lega's lab, who was at the UK when the project started, but now he's positioned at St. Jude Children's Hospital. His lab are doing cell-based studies, and they work with animals doing pharmacokinetics, pharmacodynamics, and efficacy studies that some of them I showed. And I'm happy to answer your questions, and you have this summary as a backdrop. Questions?
So real quick, have you looked at how often you have to treat, say, the mice to get them to live longer with the treatment? Do you have to keep, how often do you retreat?
Yeah, so we haven't looked in much detail on that, but so Mark has looked, these studies, the efficacy studies, we just started them, and we looked at he looked at several doses. So at the MTD, maximum tolerated dose, their best results.
Okay.
He treats them, I believe, for 5 days, I think, once a day, maximum tolerated dose, or half of the maximum tolerated dose, there is efficacy. And I think down to 1/4 of the tolerated dose, there is efficacy.
Okay.
We can talk about details. I have them.
Sure.
But basically, these are still very limited studies.
Okay, thank you.
Holly, do you want to hold your hand up real quick? Because if you need to talk with Oleg, his tech transfer person right there is Holly Simons Parker. I think you know. Does I see a hand this way? Like,
for those xenograph studies, do you let the xenograph like establish itself for a little while before you start treating, or do you start treating with drug immediately upon xenograft?
Yeah, I think the tumor is allowed to grow up to, I want to say 10 millimeters. It's something like that. Maybe 5 to 10 millimeters, I think, and then the mice are treated. So the tumors has to establish itself. I'm not sure exactly the number.
Thank you.
On that slide of you developing a phase 1, phase 2, and then phase 3, I think you'll be able to put in your phase 1, phase 2 may be good enough for regulatory approval. So may help you out.
Sorry, I can. Yeah.
With Ewing sarcoma, there's such an unmet medical need. You'll get pediatric and orphan designation from the FDA. And I think your phase 1, phase 2 will be proof of concept and approvable. You may not even need to do phase 3.
Thank you.
anyone else? Why did I have to do a little work? Patrick had to stand still. Not fair. Other side's going to have to put some work in.
Thank you very much.